the presentation. I guess my sound is up, video is up, and you should see the presentation now. All right. So yes, I will. The presentation today will be a little bit about some results I did in the past and I did through PhenoSwitch, and a little bit of results that are actually published right now in the literature from experiments that we could have performed actually, or experiments that we are actually performing, likely experiment, but from which I cannot show the results already. So yeah, well, first, what is PhenoSwitch Bioscience? It's a Canadian CRO. We provide high quality LC-MS services, and providing LC-MS services and expertise in omics means that you need to mix up a bunch of expertise. So that's the main key differentiating factors. We are not just bioanalytical chemists. We are biochemists, pharmacologists, microbiologists, people with cancer background. So we can better understand your needs, and we can align the preparation, and we can align the analysis with your needs. So this is our mantra. We try to innovate as much as possible. We are not just providing standard services. We try to work with people in a collaborative manner. Since LC-MS is quite complex, we try to simplify it as much as possible every steps. And we take great care of quality, because without quality, you cannot provide omics, reproducible omics analysis. So it's very important. We're based in Sherbrooke, Quebec. We are renting a building of 5,000 square feet. Combined expertise of more than 25 years in academia and biopharma research, 5 PhDs, 2 MSCs, 1 baccalaureate, and 2 lab techs. And we run latest state-of-the-art LC-MS instrument. So enough of description about PhenoSwitch. The other reason why I speak about this coronavirus thing is that during my PhD, I had the opportunity to actually study similar viruses, and I actually had done some experiment on coronaviruses while it was not very popular at this time. So the idea of my PhD was to study actually host cells' response to infection and the role of some endoproteases in that response. So we'll come to that a little bit later in the presentation. Yeah, today's goal. I would like us to have a bit some fun. Well, if fun, it can be about the COVID-19 publication. What is the state right now of the COVID-19 publications? I would like to highlight a few interesting COVID-19 publications that use LC-MS, and third, how we previously used LC-MS to study important viral gain of functions related to host cell proteases. So let's look at those publications. So right now there are over 900,000 publications this year to date. Checked yesterday. Over 30,000 of those publications are about COVID-19. Still, if we look at some important keyword like inhibition or inflammation, it only accounts for roughly 1,000 publications each, 500 for inhibition. And if you look at genomics, there's nearly 1,000 publications. And if you look at transcriptomics, proteomics, single cell, mass spectrometry, omics, then you see that it goes down and down. There's not much of those publications yet. So what are about those 30,000 publications? I did some text mining on the abstracts, and well, actually 30,000 publications actually tell us that I actually removed COVID-19 and redundant keywords like stress from that word cloud. And it can tell you that, well, COVID-19 is a disease that affects health, and it's severe and can or may do something. So it's not very precise if you look at the abstract. A funny thing is that if I look at the abstract at specific keywords, even though I look at proteomics or inflammation, the abstract still tells us pretty much the same thing. So I think people in the research area should be a little bit more informative in their abstracts, because they tend to emphasize too much that it's a severe health issue, and that's the main keyword that you can see from abstract. So I decided to do the same work but in titles, which proved to be a little bit more informative. If we look at the titles and we look at the last 10,000 titles published about COVID-19, you realize that most of those papers are not, I will not say not scientific paper, but more oriented to describe the disease, talk about protocols, talk about case studies, talk about patients, but they're not providing much help in understanding the disease itself. If we look at specific keywords, for example, if we look at papers published for proteomics or for genomics, we realize that we start to be a little bit more pinpointing something. So all the genomics paper tends to look at the evolution of the virus itself. As you can see, it's speaking about phylogenetic analysis. It speaks about countries where the virus actually have evolved, and about some genes in particular that present evolution. If we look at proteomics, then we start to look at something more about mechanistic. So we look at diagnostic accuracy. We look at human profiling. So that reveals us that omics research using LCMS actually pinpoint to more mechanistic review and trying to understand the disease. And that's what I want to show you actually today. So I've highlighted actually 3 papers, and the most astonishing part of this is that those 3 papers have been published in those 2 in Nature on top there, and this one in Cell. This one in July 9, so a little bit more recent, but those 2 have been published in less than roughly 3 or 4 months in that case, after the onset of COVID-19, where people started to agree that it was there and it was getting a pandemic. So if the first actually paper that we see is dated on December 31st. So it's very fast. Some of those papers have been published by very large groups, but still it has been very fast to get very strong answers. So let's look at those papers a little. So the first one I will present you is a very simple approach, affinity purification combined to LC-MS/MS, to rapidly identify all cell virus interactome. And so the workflow is pretty simple. They actually use a cell-based assay. They have cloned all the virus's gene, overexpress them with a tag, perform to affinity purification, and look at the protein that were interacting with the host cell protein. Host cell protein that were interacting with the viruses. And using that, gene ontology and connecting to database, they are able to map interaction of each virus's protein. So this is an example, and actually rapidly find known drugs that are inhibitors of those virus host cell proteins that are already published. Some of them are already approved. And that's very interesting because I think it's a very interesting take-home message from those proteomic experiments. So using a single proteomic experiment, instead of trying to screen libraries and libraries of drugs on a model using infection models, they are able to actually extract information from the also interaction with the virus, assume that this interaction may be important for the virus function, and then just use bioinformatic means to find new drugs or already published drugs that can be potential inhibitors. Second paper actually used some problem with the animation, so my my punch is toasted. They use a simple proteomic and transcriptomic approach, so multi-omic approach. They use a deep gene ontology analysis, and they actually connect this gene ontology analysis to drug target database. So the workflow in that case, they actually try to correlate transcription of a rate of transcription during the infection of viral RNAs, and they try to correlate this with the rate of transcription of the protein RNAs and the protein expression themselves. So doing this, the idea is that if it's co-regulated, upregulated at the same time, it means that it's very important for the virus propagation and transcription. And doing this, they are able to actually extract information, do the gene ontology. This is actually a sub-network because they were a main network that was related to transcription, obviously. But looking at the sub-network, they have been able to actually identify important proteins involved in the nucleotide metabolism. And they have been able to identify a known drug that is actually, I think, in clinical trials for other viruses. And they have been able to test it, and it was efficient in vitro on cell-based models. So this study actually has been received 27th of February, published roughly 3 or 4 weeks later. And it's a 2-3 months of work that has been done very rapidly to identify potential new drug for the COVID-19. So these two studies were more focused on identification of potential drugs for the COVID-19. The last one is probably my favorite. This is a kind of study I like a lot, and we like a lot to do that kind of study. So the idea was to use high-quality multi-omic analysis of serum to build a predictive classification model for disease severity. So that's one of the main problems with COVID-19 right now. Patients get admitted to the hospital, or they are positive for COVID-19, and it's very difficult right now to classify patients and know which one will be very severe or not. And that's a problem. Who will get the automatic respirator? Who will get all the healthcare that is necessary, and who actually doesn't need it at all or will not need it in time? So the interesting thing about this study is that it highlights the power of mass spectrometry. It generates a lot of data rapidly. So using multi-omics, proteomics, and metabolomics, they only use that number of patients. It's not a very high number. And they have been able to use a random forest machine learning model on those data to make a predictive classification model. And the predictive classification model, yes, it generates a lot of data, 800 or so proteins, 900 or so metabolites, but from those, actually only 29 metabolites and proteins in the serum are able to build a classification model and actually kind of predict if a patient with severe or will be severe or non-severe COVID-19 patient. So, and we are actually working on that kind of approach with clients. It's very interesting science. The other part, sorry for the change in layout. So this is from another presentation I did earlier, but this part is more about a little bit more about science. Is some stuff that I've done during my PhD and later in the company as well. So one of the key druggable target, I would say, for COVID-19 and all other virus is the entry. So how it enters the cells. It uses a receptor, and the receptors needs to bind. So the glycoprotein on the virus needs to bind to a receptor and enter the cells. But that's not the only thing that needs to be done. Pretty much as if you will see the key as a receptor and glycoprotein. Actually, it's not able to turn the knob on the door without being actually activated. So there's an activation loop on most glycoprotein, viral glycoproteins that is there that needs to be actually cleaved by also proteins. And this cleavage allows to enter the cells, so for membrane fusion. And there's an important gain of function in highly infectious viruses like H5N1, like SRAS1, SRAS2 also has it, MERS has it. It's a new cleavage site that doesn't bear only one arginine, but it's a polyarginine and polylysin cleavage site. And this is known to be cleaved by many proteases, but one in particular is the protein convertases. And this is what I've done during my PhD. And furin is the most known convertase in that family of enzymes. So what it looks like for SRAS, actually SRAS, the publication have shown that there's a spike protein, but also there's another protein in the nuclear, it's an NS1 nucleocapsid protein that also needs to be cleaved to be active. The basic model is that TMPRSS protein, in case of SRAS, is most known as TMPRSS2, will cleave at the surface to allow entering of the virus. But what we know actually is that other proteases are important. You see furin there, and goes down to infection and release of the genomic material from the viruses. What is not shown there is that, yes, TMPRSS2 is a cell surface express protein, but furin is mostly expressed in the Golgi. And what it does is that actually will pre-activate the virus. So this has been published, so it's well known. So not only the virus needs to be activated to enter, but sometimes it will just be shed pre-activated. What it does is that it's one of the key factors for systemic infection. And we are starting to know that COVID-19 is not only a pulmonary disease, it's actually infecting other part of the body, and it's probably the main problem with that virus. So if we look at the evolution of SRAS viruses and coronaviruses, you can see that there's actually two cleavage sites on the spike protein, and there's a gain of function with the polybasic site there, and the polybasic site there that could be cleaved by protein convertases, but also cleaved by other proteins. So that's one thing. So how we study this? So the main way and what people does is that there's indirect co-expression and co-localization. So this protein is expressed in the lung, and this glycoprotein is also expressed, which are the enzymes that are co-expressed. So, and there's a high probability that this enzyme will have an effect. The other way is to predict using bioinformatic models, but up to now, the bioinformatic models for enzymes are not very good. They're not built the right way, I would say. Some people have been doing 3D modeling, docking of protein docking to see if it works. The other way is to do direct analysis using Western blot. So, but you can imagine that doing many samples is quite lengthy and difficult, and the availability of good antibodies for spike proteins for Western blot, it's not there. The other way is in vitro using direct peptide cleavage, and this is what I will talk a bit about. So the most used approach to study enzymes is to use a FRET peptide with a small peptide, and when it's cleaved, well, you will lose the fluorescence or change the fluorescence, the color of the fluorescence. This allows you to do high throughput screening and rapidly identify and look at Km, Vmax, and Ki of inhibitors. The problem is that this peptide is not quite representative of what is actually happening on the protein. So the peptide is much longer, its structure as well. Some RMN experiment and crystallography have shown that in some proteins, the loop structure is pretty stable. In other proteins, it's moving a lot, but still there's some kind of a structure there. If we look at the long sequence we have there, sometimes it's very difficult to do a thread peptide. The other thing is that if you look at this peptide, there's two cysteine here that actually are able to form a cyclic peptide. And if you look at the structure there, it's most likely that it happens. So it's hard to do a thread peptide that way. It's possible, but it's difficult. So the way we were doing it in the past is by HPLC. Well, it's quite lengthy. If we look at the run time there, it's 60-minute run to be able to separate the precursor from the two fragments. It's not a very nice way. Well, it's workable if you have a lot of HPLC and you can run it. You can probably transfer that to UHPLC and reduce the run time, but still it's UV. And it works in vitro, but once you get in vivo, it's not possible to do that. What we've done actually is we've transferred that assay using LCMS actually with a 2.5-minute runtime. The separation is not necessary because we are able to select it by mass. We are able to actually monitor the cleavage, but also identify the exact cleavage site, because if you look at the sequence, there's several place that it could cut in that multi-basic site. So that's something very interesting. And we are able to actually trace Km, Vmax, and compare different enzymes and look at their role during the infection. So the study we've done with that is the following. So we tested several enzymes to cleave those peptides. And two key parameters we are able to measure is the affinity and the capacity, which are two different parameters that are important at different stages of the infection. So the affinity will be how efficient the enzyme is able to cleave and allow the virus to enter a cell at the beginning of the infection where there's not much of the virus. The capacity, however, is on the contrary, is when the virus is there at a very high level, which enzyme will allow the virus to spread more and more and more in the body. And if we look at different enzymes, we can see that all the enzymes with the different mutants of the peptides we see there don't have the same affinity and don't have the same capacity as well. So it's very tricky if you look at this. If we look at the sequence there of the mutants, they are all very, very likely, but a single modification there will affect how one enzyme or another will be able to activate the virus. And the other thing is that they don't have the same cleavage site always. So there's some kind of a backup strategy used by the virus so it can actually still enter. And this is this different cleavage site is still not known whether it has an effect on the capacity of the glycoprotein to enter to make the virus enter the cell. So that's a very interesting part. So all of this was in vitro with the peptides, incubated with the enzymes. But what we do to do in vivo or on a tissue or on a cell line, and we want to look at the protein themselves. So we do have a workflow for that. So if we look at the unactivated protein, what we do, we do chemical labeling, and using mass spec, we can actually monitor that chemical labeled peptide and monitor the entirety of the proteins. So that's a very powerful technique. And moreover, when we do that, we can actually monitor other proteins. So we can mix up this approach with a proteomic approach, with a global proteomic approach. So we can actually monitor the virus entry, glycoprotein, viral activation, plus we can monitor the whole proteome at the same time in the same method if we do a longer method. And we can actually do a targeted mRNA method and end up with a workflow as quick as we have seen for the in vitro study with a, let's say, 2 to 5 minutes gradient time per sample. So that's pretty powerful using mass spectrometry. So what I would like you to remind is that, well, LCM, SMS, omics can provide rapid and meaningful insights on the disease model that sometimes people don't think about it, but mass spectrometry is very insightful method, an insightful technique that you need to combine actually with biological, molecular biology first to adjust, like we could do to monitor activation of a protein. And also there's a big part of bioinformatics that come after that is quite necessary to actually fetch as much information as you can. And all those studies you've seen will not be possible without a robust and high quality monitoring of your instrument. So that's very important. You cannot build a multi-omic experiment with proteomics and metabolomics on hundreds of samples without a robust monitoring. Otherwise, your random forest model will only tell you this is the beginning of your study and this is the end of your study, and that's it. You will not have a very meaningful model in that case. So that's it. If you have questions, I will be glad to answer them.
Anyone has any questions for Hugo, you can type, or as he shared, you have his email address, sales at PhenoSwitchBioscience.com. He is one of our event sponsors, and we will also hopefully be seeing him in our October fall summits as well. So, yeah.
Hugo, that was a good presentation. I wondered, have you been able to share any of this data with any government agencies that are spending a lot of time and money right now looking for ways to deal with the COVID-19 pandemic?
Yeah. So right now, the cleavage assay has been done previously. You see, there's no STRAS2 peptides there. This was done with a scientist nearby us in Montreal, and is working right now with the government for that locally. And also we are doing proteomics experiment with some scientists on the US side as well, and they are sharing their data with government agency around that. And I think most of the paper I've presented, they have made their public open source, their data open source. So it's easy to access all that information and fetch that out. So it's very interesting for pharmas and biotechs who may have a lead that target those proteins or pathways. I think it would be very interesting for them to just look at this and see if it could fit.
Well, that's good, because I know that there's a lot of activity and a lot of people interested. So it's good that you're able to provide some additional insights on how we might address the pandemic.
Sure.